Hello, everyone. My name is Jordan Mulliken, and I am currently pursuing forensic biology through the George Mason University Forensic Science Program. I am honored to introduce to you forensic examiner Amanda Bakker. Ms. Bakker is a forensic examiner in the DNA casework unit of the FBI laboratory. Starting her career with the lab in November 2007, Mrs. Bakker has held multiple positions all within the DNA units. From January 2009 to March 2015, she was a biologist within the DNA unit, processing items of evidence for serology and DNA analysis. Since March 2015, Mrs. Bakker has been a DNA forensic examiner and is qualified in DNA analysis to include statistics using probabilistic genotyping. She has been qualified as an expert and has testified both in state and federal court over 20 times. Now, before I bring her up here, I would just like to say we will have volunteers with microphones for the Q&A section. So if you're going to ask a question, just hold off for 1 sec for the volunteer to get to you so that we can all hear you clearly. And with that, I would like to introduce forensic examiner Amanda Bakker. Thank you.
Thank you so much for that introduction. Good afternoon, everyone. Thank you for having me. It is gorgeous out today, and I was very happy to leave the lab. So like Jordan said, my name is Amanda Bakker. I am an examiner in the DNA casework unit at the FBI laboratory. So before I begin, I would like for you guys to humor me for 2 seconds. I did this at my son's elementary school career day, and it went over really well, so I think it'll go over well for adults. So if I can have everyone stand, please. It won't be for long, I promise. All right, now that we're all standing, if you have brown hair, can you please sit?
What about brown and gray?
That's you. If you have blue eyes, please sit. And lastly, if you have wings, please remain standing. Wings, like bird wings. Not a trick question. So based on our DNA, we could answer those questions. Our DNA gives us things about us, our hair color, our eye color, as well as the fact that we don't have wings and we don't have tails or scales. So thank you for humoring me. I did do that in front of a bunch of kindergartners through 5th graders, and I always had 2 that remained standing every single time. So what we're going to talk about today is DNA. What is it and what do we do with it? This is our standard thing at the beginning. I'm going to talk about different kits that we use, different probabilistic genotyping software. That's what we use within the laboratory. Please know that there are other kits and probabilistic genotyping software that you can use. I'm not endorsing any of them. I'm just letting you know what we do in our laboratory. So this is a very long list of learning objectives. I actually gave this presentation about a month and a half ago at the National Advocacy Center down in South Carolina. So what I did is I merged the DNA and the statistics presentation together. So hopefully it's not too long and too boring. I love science, so I'm very passionate about it. And one question I actually meant to ask, who here are lawyers? Okay, any law enforcement? And any students or academia? Okay, just so I know who I am talking to. At any time, I like to keep this pretty informal. If you have any questions, please don't hesitate to ask. But again, like Jordan said, we can wait till the mic gets to you. That would be awesome. All right, so what is DNA? DNA stands for deoxyribonucleic acid, and it's our genetic makeup. We inherit half of our DNA from our mother and half of our DNA from our father, and it makes us who we are. So again, it gives us our hair color, our eye color, and it makes sure we don't have things like wings and scales. Over 99% of our DNA is the same. That makes sense. If you look around the room, we're all humans, with the exception of identical twins who share 100% of their DNA. So if I were to take a DNA sample from a set of identical twins, I wouldn't be able to tell them apart based on their DNA. Less than 1% of our DNA is actually what differs amongst us, and that's what we focus on for forensic purposes. We call it non-coding DNA or junk DNA. Essentially, it's not coding for anything. So when I'm looking at DNA for forensics, I can't tell right now if someone has blonde hair or blue eyes, if they are susceptible to any kind of cancer or any other medical abnormality. We don't look at that within the laboratory currently. And there are two sex chromosomes that we look at, X and Y. Females have two X's and males have X and a Y. Where is it found? So it's found in the nucleus of the cell. So if you think of a cell like like an egg, the nucleus of the cell is equivalent to the yolk of the egg. That's where I'm focusing on nuclear DNA. So we'll talk about a couple different other types of DNA. Every cell, so if I were to take a blood cell from you and a skin cell from you, a swab of the inside of your cheek, your DNA is going to be the same across all cell types. There are some caveats to that, but I won't get into that. That gets really into the weeds. So generally, our DNA is the same across all cell types. And it's the same throughout our life. So the time you're born to the time you die, your DNA is the same. What is our goal in DNA testing for forensics? It's essentially to develop a DNA profile that we can compare to either a known individual or to other samples. And a DNA profile just looks like a string of numbers, and I'll talk about that more later. So if I have a sexual assault and I have a vaginal swab and I have a profile from that vaginal swab, I want to be able to compare it to a known potential perpetrator or even a consensual partner to give you information on, does this match the individual you think or does it not? So what can it tell us? DNA testing can tell us, is biological material actually on an item or an object? We can also determine, is it human? So our DNA testing within the laboratory is human specific. I can tell the biological gender of an individual, and I can tell who that biological material belongs to. So again, if I were to swab the inside of my cheek, process that swab for DNA, I know that that DNA profile is special to me. What it cannot tell us. This is the who, what, why, or when. It can't tell us how it got there, when it got there, how it got there. It can't tell me, was it on an object through direct contact or through transfer? And we'll talk about that a little more later. Are there different types of DNA testing? So spoiler alert, yes, there are. The first type is nuclear DNA. So again, this is half from mom, half from dad. We're all slightly different with the exception of identical twins. Most of our cases undergo nuclear DNA testing because it's the most discriminating. We want to have the best amount of information. And again, the non-coding areas. So if you follow this tree, so the squares are men and the circles are women. So if I'm here, let's do here. This would be my dad, this would be my mom, this would be me, and then this would be my child and my husband. So that's how nuclear DNA is passed down. Y chromosome testing is passed down through the Y chromosome, unchanged from father to son. So anybody in the same paternal line, barring mutations, will have the same Y DNA profile. So again, if I were to use the example, if let's say this is my husband here, his father, my father-in-law, and my two sons all have the same Y profile. A lot of times we use Y testing in sexual assault kids, sexual assault cases. We have so much female DNA on body swabs that we essentially want to target that male DNA. So we'll perform Y chromosome testing. And of course, mitochondrial DNA, that is passed down from mother to child unchanged. So my mom, myself, and my two boys all have the same mitochondrial DNA, but since they are boys, it will stop with them. They will not pass it on to any future offspring. It's, again, very similar to Y's, barring mutation. Everyone in the same maternal line will have the same mitochondrial profile. Again, it's not as discriminating, but it is helpful. A lot of times we use these in missing person cases. So if we have a body or hairs that are really old or have been exposed to elements, the nuclear DNA might be broken down and we can't get a lot of nuclear DNA, so we'll try mitochondrial testing. So what are we testing for in DNA? We're testing something called short tandem repeats, or STRs. And these are small segments of DNA that repeat themselves over and over again. We all have the same sections of DNA, but the amount of times that they repeat is what's different between us and what we inherit from our parents. So for example, at one small section, I may inherit 10 repeats from my dad and 11 repeats from my mom. So my DNA type at that particular location would be a 10-11. Somebody else in this room may have 18 repeats from dad and 21 repeats from mom. So their DNA type would be 1821. Where do we get DNA from? Everywhere. DNA can really be found on anything that's come in contact with an individual, whether it be directly or through some kind of transfer event. Typically, we look at blood and semen stains, and we also do what we call wearer or handler, and that's trying to collect skin cells from an item of evidence. Some of our evidence goes through serology, and this is identifying body fluids on items of evidence. So for example, if you have a sexual assault that took place on a king-size comforter, we don't want to have to take a Q-tip and swab the entire king-size comforter. So what we're going to do is we're going to take a look at it and see, are there any stains blood or any stains semen? And if so, we'll focus on those first because we know those fluids are rich in DNA. This is not an all-inclusive list. Pretty much any item you can think of, we can get DNA from. And we just talked about forensic serology. Profile again, so it's a graph that represents an individual's DNA types. So it looks like peaks on a graph, or I like to explain it as spikes on a heart monitor. Ideally, an individual's DNA profile will have 2 peaks at each location, one from mom, one from dad. Now, if they inherit the same number of repeats from both mom and dad, you may see just 1 peak, and I have examples of that. In an ideal world, again, those 2 peaks are going to be at equal heights because we're expecting the same amount of DNA to be passed down from mom and dad. And again, like I mentioned before, we're also looking at 3 sex determining regions to let me know if a profile is from a male or a female. So here, again, a DNA profile. So we use the KIT Global Filer. We look at 21 locations on an individual's DNA, including those 3 sex typing locations that I mentioned. So this one and this one and this one are all sex typing. This is a single source profile from a male, and that means it's from one individual. So as you can see, most of these locations have 2 peaks. When I talk about locations, it's each of these rectangular bars. So this is a location, this is a location, this is a location, and so forth. Locations, for example, like this, that individual inherited the same number of repeats from both mom and dad. Female profile, single source, very similar with the exception here and here. These are the 2 locations that let me know if the profile is from a male. This is a female, so I don't see anything at those 2 locations. How do we develop a DNA profile? So this is done in the laboratory. There's 5 steps. The first step is collection. And this is where we're going to take a swabbing or a cutting of an item of evidence. We're going to place that cutting or that swabbing into a tube. And essentially what we're trying to do is we're trying to remove any cells that are left on that item, whether it be skin cells, blood cells, or sperm cells. And then the next step is extraction or purification. And this is where we want to break open those cells. So again, if you think of my example like an egg, we want to crack open that egg to get to the yolk or the nucleus of that cell. And we want to wash away all the pieces we don't need. So the egg white, the shell, any other proteins that are there, we want to get rid of those and keep just the yolk, or again, the DNA that's in the nucleus of the cell. This is done mostly by robotics. So a biologist in the laboratory will actually swab the item of evidence. They will cut that swab and place it into a tube. They then add chemicals to that tube to start the extraction process. But then that actual process of cleaning and purifying is done on what we call a robot or an instrument. And here's an example of one here. The next step is quantification, and this is determining how much DNA we have in our sample. So again, that small tube, we start with the swabbing, we add chemicals, we have such a small amount or a very small amount in the bottom of the tube. We then put it onto another instrument to let us know, do we have a lot of DNA, do we not have a lot? And this also is our first indication if we have male or female DNA. I also mentioned batches and controls. So at the FBI laboratory, we get a lot of cases. So it's a lot of samples. So we actually do batch our samples from different cases onto one instrument. That might cause a red flag for some of you in the room. We validate it. I'm going to talk about that a little bit later. But one thing maybe to keep your mind at ease, we also run controls with all of our processes. And this is a known profile that we know what the answer should be, and we send it along with all of the other samples to make sure that at the end, we're actually getting that known profile. This is making sure our instrumentation is working appropriately. It also makes sure that our chemicals are working appropriately. On the flip side of that, we have a negative control. So this is a tube that doesn't contain any DNA at all. And we want to make sure at the end of the process, we still don't have DNA. That's going to make sure that there was no contamination introduced via our chemicals or our instrumentation or our processing. A fourth step is amplification, and you can think of this like genetic photocopying. So we're taking those segments of DNA and we're making millions and millions of copies of them so we can actually see them. Again, this is done by robotics, and again, we batch our samples, but we do have controls. And then the last step is separation. And this is where we're taking those pieces of DNA and we're separating them by size so that we can ultimately get that DNA profile that I showed you before, those peaks on a graph. Again, robotics, batches, and controls. So what type of testing has been done at the FBI? Some of this is pre-me being an examiner, but here's a list of how things have kind of progressed throughout the years. We're at Global Filer right now, which we look at 21 loci. Our previous kit, identifier, looked at 15. So really that's just, the previous kit was not wrong. We're just getting more and more information. So we're able to get better statistics, which we'll talk about later. So anything that was processed pre-2017 with identifier is still good to go. Just know that now we have a little bit more information. We're a little more discriminating, which is going to make your statistics higher. So why do we change anything? I jumped the gun. More discriminating power. It also helps us get better data from degraded samples. So some of the kits that we used previously, without getting super scientific, it was looking for some of those locations that were really big. Because we know when DNA breaks down, basically what that means is an imaginary scissor is coming and cutting that DNA into pieces. Once that happens, our DNA profile is not good. So as the kits increase, so does the power to amplify degraded samples so that we can use the data. We can also combine kits. Prior to identifier, we actually had profiler and cofiler. Those were two different kits, and they looked at two different segments of DNA with a few overlapping, so we can make sure they were concordant. But that took time and resources to run each sample with two different kits. It essentially took twice as long. So now with Global Filer, we've put all of those locations into one, and we can do things a lot faster. So how I interpret a DNA profile. So once I get a profile, at the end of that whole process in the laboratory that I had mentioned, I first determine, is this male or female? So I take a look at those 3 sex determining regions and determine, do I see stuff there? Is it male? Is it female? Is it potentially a mixture of both? And I'm using our standard operating procedures to help me interpret profiles. I also look at the profile and determine number of contributors. So again, if you remember, 1 peak from mom, 1 peak from dad. If I have a location that has 4 peaks, that gives me an indication that I may actually have at least 2 people there. So I will look at the profile as a whole to determine how many people I think make up that profile. And then sometimes we do get artifacts. Sometimes samples, we see bacterial peaks. We have those documented in our standard operating procedures as well. So I take a look at our profile to determine, is this all what I think is human DNA, or do I have some of these artifacts that I can cross out? Right, so I think I showed you this one once before. So this is an example of a single source male profile. There's 1 or 2 peaks at each location, and there is data at all 3 of these sex typing locations. This is where it gets a little more complicated. So this is actually a mixture. So this is more than 1 individual. Looking at it quickly, I would say this is probably 2 people. I actually know the answer is 2 people for this one. But again, I'm looking at how many peaks here. I have 4 peaks at this location. Here I have 4 peaks. I have some information at all 3 sex typing locations. I know it's a mixture of male and female because of the imbalance between the X and the Y. And again, I'm using my protocol to help me determine if it's male and or male and female. So I look at both the graph and I also put it in a table formula or format, excuse me, so that I can easily compare any known samples that I have to my evidence profile. We won't go through this in depth. This was at the NAC, we actually had them go through and compare things. But since I'm limited to time, I will give you the answers. So a male profile and a female profile, these were reference samples taken from an individual. So again, that can be from a cheek swab or a blood sample. I know that if I swab my cheek or I take a sample of my blood, the DNA that I get from that sample belongs to me. If I were to swab anybody in this room and process your sample, that DNA profile would be for you. So we refer to that as the known sample or the reference sample. It should always be single source. So if I swab my cheek and I process it in the lab and I don't get a single source, something's wrong. So usually I go back and look at, was there a sample switch? I hope not. Or was it a situation I've had samples in the field that have been switched? The same nurse took a sample from the female and also took a vaginal swab and swapped them. So the vaginal swab came up as single source, but then the female swab, the known reference sample, actually came up as a mixture. So I use that to help me determine, do I have the right sample? Is it processing appropriately, or do I need to do a little more digging? All right, so I've developed a DNA profile and I have samples to compare to. Now what do? I have to draw conclusions. So first I can determine, when I compare a known sample to an evidence sample, I can determine that someone is excluded. And this means that the DNA from that known individual is not present or is not represented in my sample from the evidence. I can make the determination that that known individual could not have left that particular DNA behind. For an inclusion, there are some similarities. So the known reference sample matches or almost matches my profile, my evidence profile. So when I determine that, I can say I can't exclude them. Maybe there's an inclusion. All right, so this is an example of that single male contributor, which matches to this male contributor here. If you were to compare each of these peaks to each of the numbers here, for example, so there's a 15, 16. I know he's a 15, 16. I would do that throughout the whole profile and determine that there's potentially a match. So I have a match. Now what? Statistics. I heard the presentation prior to me talking about statistics. People either love it or hate it. It's okay. Once I determine that I have a match, I do have to calculate a statistic to give weight or significance to that match. So this is another example of the male and the female mixture. I compare the male and I compare the female. And what conclusions can I draw? I can say that the female is included because her DNA types are represented in that mixture. So for example, here's the mixture here. My female is a 1618 for the top location. There is a 1618 in the mixture, 1619, 1619, and so forth. So I can say that she's potentially included. The male I can exclude, and I'm actually going to jump to the second one. So this male individual is a 1416. I have a 16 here, but I do not have a 14. Because I know this is a two-person mixture, I have 4 pieces of information. So one from mom, dad. So because this profile does not have a 14, I can say that this person is excluded, meaning he's not included in that mixture. And I'm going to skip that. So why do we do statistics? Not all matches are created equal. I like to use the phone number example a lot, especially when I'm talking to prosecutors or agents in the field. If I have one digit of a phone number, actually, we'll do another little activity. If you have a 5 in your phone number, raise your hand. Okay. If you have a 5 and a 2, keep your hand up. A 5, a 2, and a 1. So as you see, I won't go through the whole phone number, but as you can see, as I got more and more information, we were able to cut down people in the audience. DNA is very similar. If I have one peak at one location, probably half of us in this room will match. As I get more peaks and more information, I'm slowly going to have people sit down until hopefully there's one remaining standing. A person's complete DNA is unique to them. I'm going to caveat that, but we only test 21 locations. Why I caveat that is statistically and scientifically, I don't expect to swab anybody else in this world that has my DNA profile. However, I cannot swab everyone in this world. So it's very possible that someone on the other side of the ocean in some small little town has the same DNA profile as me, just based on how they've inherited their DNA throughout generations. We don't think that will happen, but we can't be 100% sure. So you will never hear me say it is a 100% match. Nobody else is going to be included, because we can't say with certainty that no one else in the world has the DNA profile. And remember, identical twins who actually do share their DNA profile. So it is not unique to each of them. It's kind of unique to that group. Population genetics tells us that a 21 locus profile is certainly unique, but again, I don't know that for sure without testing everyone that's living in the world, who has ever lived and who will ever live in the future. Not all evidence gives us a 21 locus profile. So I had just talked about that with the phone number. Sometimes we get peaks at every location. Sometimes we only get 1 or 2 peaks. So based on the statistics, you're not going to have someone match really, really, really well with only 1 peak. But the more information that you add, the better I would expect my statistic if they are a true match. The more complete the profile, the less chance there is to match by coincidence. So again, going back to the phone number, if all I were to tell you was that the phone number I'm thinking of has a 5, half of you in this room matched it. As I added more information, fewer and fewer people matched that information. And this is why we give a statistic to give weight to that match. All right, so another little activity, I won't make you stand. I'll have you do this one in your head. So everyone think of a car at random. It doesn't matter. You don't have to do this car. This was just, my kids were in my head when I did this, but think of a car. All right, now, if you thought your car, the car that you thought of, if you thought it was white, 19% of US cars are white. So if you didn't think of a white car, you're out. We're going to add more and more information. So adding 2 doors, adding that it's a Volkswagen, it's a Beetle with round headlights. Did anybody think of this car? Probably not. The last group I offered to buy them lunch, even though lunch was provided. But so this is why statistics are needed. So again, as you can see, the more information we add, the smaller and smaller of the pool of possible matches we can get. And that will be portrayed in our statistic. Basics of statistics. Each allele has an estimated frequency in the population. So we have taken samples from known individuals, and we look to see how often does a person inherit 15 repeats at one location. We do that across the entire DNA profile to give us an estimate of the frequency of alleles. We then use these allele frequencies to determine locus frequencies. And each locus is independent of the rest. So what I mean by that is what we inherit here has nothing to do with what we inherit here. And that's the same across all 24 locations. And then what we do is some special math. We multiply all the frequencies together, and that helps get our statistic. Population genetics is the core of all DNA statistics. So again, in order to figure out frequencies and how great this matches, I have to know an estimate of what I'm looking at in the population of the world. And there have been studies to show the frequencies. Some are more similar than others based on populations. So we have Native American populations. We know based on population genetics and statistics, the individuals in those groups tend to not have as much diversity in their DNA. We take that into account when we look at our population frequencies. So there are differences across populations and even within populations. And these frequencies are published. They're in our standard operating procedures, and you can also Google them. They are published and well known within the community. So traditional statistics prior to 2016, I believe, we used to use RMP and CPI. Who here is familiar with RMP and CPI? Okay, a couple. So RMP was the random match probability. And this told us how many people in the world would I expect to see this. So a lot of times your statistic would say 1 in a billion or 1 in 500 trillion. And we would extrapolate that out to how many worlds would I need. It's not quite how we do things now. I'll talk about that in a second. And then the combined probability of inclusion was slightly different. We use that a lot with mixtures. Again, looking at frequencies to see what's the probability of this person being included in this mixture. This was threshold based. And what I mean by that is during our research, our validations, we know that DNA has to meet a certain threshold for us to be confident that we see all of the information. So for example, here, I see one peak and I will tell you that it falls below our threshold. So I don't know, does that individual truly only have one peak at that location? Or did that second peak drop out, meaning that there wasn't enough there for me to detect it? So using our previous statistics, we had to throw away all of that information. If we weren't sure if we were seeing everything there, we couldn't use it in our statistics. Again, the way we did things before was not wrong. It just wasn't as informative, and we got rid of a lot of information that we could have used that we can actually use now. The previous comparison that we had, the female would be inconclusive because I can't tell based on the limited amount of DNA here. Is she potentially here? Is she not? I can't include her. I can't exclude her. So she falls in that inconclusive range. Who here is familiar with probabilistic genotyping and likelihood ratios? Who here has tried a DNA case? Okay, that helped. So likelihood ratios is actually how we perform our statistics now. We use probabilistic genotyping, and we use a software called StarMix. There are other softwares out there. TrueAllele is one. I think I have a list on the next page. This allows us to use all of the information that we see. So before, I had to throw away some of these locations that only had the one peaks because I wasn't sure, am I seeing all of the information? Now using probabilistic genotyping, I actually can use all of the information. The StarMix was developed in response to actually a laboratory using CPI incorrectly. So they weren't getting rid of some of that information we didn't have all of the information of. So it was developed by ESR, and it was first used in 2012 in casework. Now there's over 80 labs in the United States that use StarMix, and 9 states and territories of Australasia and 16 additional labs. I don't know off the top of my head how many are validating it currently, but a lot of laboratories are going the probabilistic genotyping route. And like I mentioned before, at the bottom here, there are other software systems that we use. So what is a likelihood ratio? A likelihood ratio is comparing the probabilities of evidence under one of 2 different explanations. So the first explanation is that an individual is a possible contributor to that DNA. And the other explanation is that an unknown, unrelated individual is a contributor. Generally, whichever scenario has the higher probability gets the weight. And the significance of that weight is determined by the ratio of those 2 numbers. And if your eyes did not roll in the back of your head, I kind of hope they did. But so to give you a little more life example, so imagine I have 2 bags of marbles. In one bag, I tell you that I have 10 marbles. There's 1 red and 9 blue. And in the second bag, I tell you that I have 100 marbles, 1 red and 99 blue. I tell you that I've drawn a red marble, but I haven't told you which bag I've drawn from. I can actually calculate a likelihood ratio for that. So first, if we take the 10 marble bag, I know the probability of drawing a red is 1 out of 10, or 0.1. Then if I were to take the 100 marble bag and calculate the probability of drawing a red marble, I can determine that the probability is 1 over 100, or 0.01. I can use these 2 numbers, 0.1 and 0.01, to calculate a likelihood ratio. And I would say that drawing a red marble is 10 times more likely if I drew from the 10 marbled bag than if I drew from the 100 marbled bag. It's possible to draw from either bag, But the question is, which one is a little more likely? It's a little more likely to draw from the 10 marble bag. With DNA, we use numbers much, much higher. Our likelihood ratios, the highest I've ever seen is a non-nillion, which is 30 zeros. So using that population data and calculating likelihood ratios, we can get statistics extremely high. How does our probabilistic genotyping software work? It's actually a two-step process. The first is deconvolution. What the software does is it takes the mixture that I import to the software and it's going to try to separate it out into its individual contributors. So I like to use the yarn example. If I have a ball of yarn with 100 different colors of yarn, deconvolution is putting all the green ones in a pile, all the red ones in a pile, all the blue ones in a pile. That's what StarMix is doing for a DNA mixture. It's taking template and degradation, amp efficiency, and stutter. It's taking all of that into account to try to determine is all of these DNA pieces from contributor 1 versus contributor 2 versus contributor 3 and so forth. Template is how much DNA is there. So when you think back to the peaks on the graph, the profiles, peaks that are extremely high means there's more DNA, there's more template. Peaks that are really low means there's not as much template or DNA there. Is it possible that one contributor is donating more DNA than another? It's absolutely possible. It's very rare that you have a 1-to-1 ratio. Sometimes we'll have what we call a major contributor, and that would be equivalent of me donating a cup of my DNA versus any of you donating a teaspoon of DNA. So we have a major contributor. The minor contributor is the person who's donated a very small amount of DNA. Degradation, we talked about this before, how much DNA breaks up. So the software can determine based on the data that it's seeing, how much does it think that the profile is actually degrading. Amp efficiency, how well does the DNA copy? And is one contributor's copy better than another? So you could have my DNA profile that is straight across the board, template's really high, there's no degradation. If I mix that with my husband's, his may come across kind of we call it a ski slope effect, where on one side of the profile, it's very high, but it degrades and it gets lower on the other side. That's amp efficiency. We're determining, does one person copy better than others? Stutter, it's a byproduct of the DNA copying process. When I was talking about artifacts earlier, this is something under artifacts. We look at this during our validation and our testing. We know some of these artifacts appear, and we can input that into StarMix, and it will calculate the rate at which things stutter. So then we use the Markov chain Monte Carlo, MCMC. This is used during deconvolution. It's based on a lot of mathematical principles that have been around for several, probably tens, if not 50s of years. What it does is, again, without getting into the weeds, it deconvolutes the profile, and then it determines, it creates a fake profile, and it compares it to what actually I have put into the system. And you can think of it kind of like a game of hot and cold. It creates this fictitious profile, it compares it to the profile that I know I have, and it's going to say, based on the template and the amp efficiency, is this a good fit or not a good fit? And it's going to make tweaks until it gets to the best profile that it guessed that matches what I've inputted into the system. A lot of times we also think of this like when you go to the eye doctor. So there's quite a few of us in the audience that have glasses, I wear contacts. When you go and they tell you which one looks better, 1 or 2, and one's usually slightly better than the other, the MCMC process is very similar to that. We're trying to refine that profile. Some other areas that MCMC is used in engineering, weather predicting, the stock market, and more recently, I actually discovered search and rescue. So if a ship goes missing, what they'll do is they know based on the last SOS call, it's in 100 square miles. Then they'll have somebody search it. They'll search maybe the top square mile. They know they've searched that. They're going to search another area. They know now these 2 top locations is not where the ship is. So they're eventually going to hone into potentially where that ship is. So they're using an iteration of MCMC. And then the second step of STAR Mix is it performs a calculation. So once it deconvolutes that profile, it gives me my individual contributors. I can then actually use population statistics to calculate a likelihood ratio. It's using genotype weights and population frequencies to help calculate that likelihood ratio. And again, those population frequencies are what's published in the data. For all of our cases, we report the 4 main populations in the United States: Caucasian, African American, Southeast, and Southwest Hispanic. And then we also have additional population groups as needed. So if a case, and this is all based on the evidence, not based on the potential POI. So if a case takes place in the Caribbean, we will calculate a Trinidadian statistic. If it takes place in Guam, we'll add the Chamorro and the Filipino. We also have Native, Alaskan, and Native American populations as well. For Asian populations, we actually don't have a published Asian population group right now. And the reason we don't have one is we just didn't have enough volunteers. When they did this years and years ago, we didn't have enough individuals of Asian descent to volunteer their DNA profiles. But again, when I mentioned before, we've done studies to show population frequencies between and across populations. So even if a case takes place in a location that has a high Asian population, we know the other 4 main United States populations will cover those frequencies. So a lot of times I get the question, what if my POI doesn't fall into one of these categories? I get this a lot in Native American territory. We report Navajo, Minnesota Native Americans, and and I'm going to cheat because I can't remember the last one off the top of my head, Apache. So a lot of times I get a question saying, my individual is not from those 3 populations. What do I do? And I say that doesn't matter. It's all based on where the crime occurred, not the race or ethnicity of an individual. We are covering the most common populations in the United States, and we know that different subsets were compared to show there are similarities. So an individual from Cherokee Nation will have similar population frequencies to Navajo. So we don't need a Cherokee database. As long as we use the 3 Native American populations, we know we're good. We never want to assume the race of the person of interest up front. We don't want to skew our results any way, so that doesn't matter to me. One of the main reasons for that is people I identify differently, and I have a story about this that I got permission to tell. So my mom absolutely loves DNA. She watches forensic files every single day. She calls me every day with questions. She thinks she's a DNA expert, which is great because it gives us something to talk about. So because she was so excited, my brother decided a couple years ago that we were going to buy her 23andMe for Christmas so she could learn about her own DNA. I grew up thinking that I was half Italian, half German, Irish, Scottish, everything on my dad's side. Well, come to find out that is not true based on 23andMe. So my mom, if anyone were to ever ask her, she would say she's 75% Italian, 25% of another combination of things. Based on her DNA, we actually found out that her dad is not her dad, and she's actually partially Mexican. So in talking about population groups, I might say that I'm 100% Italian, which I used to tell my friends all the time when I was little because I didn't understand, but that's not right. So we don't want to rely on individuals self-reporting their ethnicity because they may not know. They may wish they were another ethnicity, so they say that that's what they are. So people identify differently. So we don't worry about what the race or ethnicity of a person of interest is. So what does the likelihood ratio mean? It can fall in one of 3 categories. So if you think of likelihood ratio as a sliding scale, and I'm going to try to do this the right way since I'm backwards from you all. So if I have a 0 on this end of the scale, that means that that individual is excluded. My likelihood ratio is a 0. I can say that that person is not included. Anything from 0 to 1, I can say that there's support that that individual is excluded because we're getting towards the 1. 1 is dead set in the middle, even though it's not really in the middle, and that's uninformative. I can't tell you is a person included or excluded. They're not giving support for either. And then the farther to the right we go from 1, that's more support for inclusion. So the likelihood ratio is a numerical value. We determine where it falls on that scale. Is it 0? Is it less than 1? Is it 1 or greater than 1? And here, actually, it's the same thing. It just goes up and down. So here in the middle, we have a likelihood ratio of 1, which is uninformative. The higher the number that we get, the more support for that initial hypothesis that an individual person of interest is a contributor. And then the farther away from 1 towards 0, we get support for exclusion, and then we get an outright exclusion. In addition to our verbal scale, we also report, in addition to our numerical value, we also report a verbal equivalent. And this is to give juries context to what a likelihood ratio number means. If I were just to testify in the stand that I got a likelihood ratio of 5, some jury members may think, yep, that's him. Some may think that doesn't mean anything at all. So we want to give some kind of context to the field. So we give our numerical value that was originally adopted by the Association of Forensic Service Providers, and actually SWGDAM, the Scientific Working Group on DNA Analysis Methods. They actually, this is now their verbal scale. So SWGDAM is a group of practitioners and academia members that come together and talk about all things DNA. One thing they did more recently is they wanted to make sure the verbal scale was consistent. So you had some laboratories that would say, for example, a likelihood ratio of 100 to 9,999 is moderate support. Some other labs may say very moderate support. Well, what does moderate support mean versus what does very moderate support mean? So we wanted to make it standard across the community. So we now use the standard scale that was published in the SWGDAM guidelines. And really, a lot of times I'll have prosecutors ask me, how do I use this? And I like to say, use it in conjunction to the number, but the number doesn't go away. So if I have a likelihood ratio of 40 quintillion, that is greater than 1 million. So it's very strong support for inclusion, but so is 2 billion. So I like to tell juries, I have this sliding scale, the higher the number, the better support for the inclusion, but to also give some kind of context, especially when I fall into ranges around here, 1 to about 1,000, it lets the jury know, is it uninformative, support for exclusion, limited support, moderate support, so forth. So what happens if we get it wrong? So forensic DNA is never exact. Even if I look at a profile and I say that it's a 2-person mixture, it could actually be, in fact, 3. I'm using my training, my expertise, my standard operating procedures to give the best, I hate to say best guess, but it's our best scientific estimate. But again, we never truly know how many people have contributed their DNA. We've looked at both developmentally and internal validations on what happens if we get the number of contributors wrong. So a developmental validation is done by the creators of the kit or the software. So thinking about StarMix, ESR was the creator of StarMix. They did a developmental validation. They wanted to make sure that the calculations worked appropriately, that the software, when you put in a text file, it didn't spit out a picture of something. And then the internal validation is what we do at the FBI laboratory to make sure it works within our laboratory. So another analogy I like to use is brownie mix. So maybe Duncan Hines is the developer of that brownie mix. So they perform their validations. They want to know, how many eggs do we need to put in? How much oil do we need to put in to make our brownies the best? The internal validation would be me baking those brownies at my house. My oven always runs a little bit cooler. So I might have to put my brownies in for 25 minutes as opposed to 21 that the back of the box says. So that would be an internal validation. I'm taking that brownie mix and making it specific for my house, my use in my house. So both developmentally and internally, we tested what happens if we get the number of contributors wrong. And we did this by creating our own fake mixtures. So we took, for example, a known profile from me and a known profile from my coworker. We mix the two together. We know this is a two-person mixture, but I'm going to tell star mix it's 3, or I'm going to tell star mix it's 1. What happens? We found that typically underestimating the number of contributor leads to a false exclusion. So if I have a true 30-person mixture, I have 2 great major people and a small minor, someone who's contributed a really low amount of DNA. If I say it's actually a 2-person mixture, that minor person is probably going to be excluded because it's not the tall 2 people that I see. Overestimating the number of contributors can actually lead to a false inclusion. So if I have a 2-person mixture and I say that it's 3, the software is going to say, well, I expect to see 6 peaks at each location. Ideally, I only see 4. Maybe we've had some dropout. Maybe some of those peaks are so low that we can't detect the DNA there. So I'm going to say that that person might be included, but that inclusion is going to lead to a very low likelihood ratio. So it's a false inclusion, but you're not going to get a false inclusion with numbers in the non-millions. Usually where that happens is like a likelihood ratio of 5, where they could be there, they might not be there. So it's, again, low level likelihood ratios. The major contributor, there's not much effect. They're either there or they're not. The minor contributor has the greatest effect. So again, if I have that low level minor contributor, I have a true 3-person mixture and I say that it's 2, that minor person might be excluded. If I have a true 2-person and I say that it's 3, that minor individual may actually be included, but again, with low level LRs. We have something called diagnostics. We think of this almost like the dashboard on your car. As long as there's no lights on your car, you're good. The minute something pops up, tire's flat, engine needs to be looked at. We have something very similar in star mix that we look at with every run. Every calculation that we do, we make sure that those diagnostics are lining up the way we want them to, that there are no red flags on the dashboard. If there are, that lets me know as the examiner, did I do something wrong? Did I put the wrong profile in? Did I say that it was 4 people when it's clearly 5? I can count peaks. I can count up to 10. It should be 5. So then what I can do is I can adjust the number of contributors. I can rerun the software and take a look to see if that helped with the dashboard. How do we know we do good work at the lab? So we've already talked about some of these. So validation, it's a process or procedure that undergoes extensive testing to make sure that it works both as a whole and within the lab. So again, the brownie mix, we want to make sure that the brownie mix as a whole will make brownies and not make cupcakes. And we also want to make sure that the brownie mix works in my oven at my house. It ensures consistency within and between cases. So every time I use a specific DNA kit and a certain amount of chemicals, whether it's from this swab or this swab, I want to make sure that I'm getting the same not data from both, but making sure that I get the best data from both. So you can think of this going back to the brownies, using Wesson oil versus, I don't even know another brand, Great Value. It's both oil. We just want to make sure that they both work in our brownie mix, and I'm going to have consistent brownies each time. We talked about the developmental, internal validations. We talked about that. Volumes of samples needed when we do validations. So we do a validation. That validation then helps us write our standard operating procedures. So that's going to tell us how much oil do we add? How long do we bake it for? How much chemicals do we use? How long do we run our amplification cycles? Based on that, we can then train our biologists and examiners based off the standard operating procedures. And then once we're all trained, we then can start performing these processes or procedures on actual casework. Accreditation and standards. So laboratory accreditation, it's ensuring that we're following standards. So the FBI laboratory is accredited by ANAB, which is the American National Standards Institute National Accreditation Board. And we are audited or reviewed every 4 years to make sure we're following their standards. Some examples of their standards may be that as an examiner, I have to hold a 4-year biology degree. So an auditor will come in and take a look at my training records. As an examiner, do I have a 4-year degree? Do I have all 4 main classes that I need? They're also going to take a look at our training records. Did I undergo all of the training that I had to in order to be a forensic examiner? How we process cases, how evidence is stored. Those are just a small subset of the different types of standards that we're audited to. There are multiple laboratories that are audited to the same standards, again, to make sure that we have consistency amongst the community and that we're doing good work in the laboratory. This is just some other standards that were audited to ISO 17025. The more ANAB is a little bit more specific. So 17025 may say you have to have 1 examiner in your lab. ANAB may say you have to have 2. So it's the further down in the standards that you get, the more stringent it gets. Then we have our quality assurance standards. So those are very specific for our DNA laboratory. So ISO 17025 may say you might have to have 1 DNA examiner. ANAB may say you have to have 2. Quality assurance standards may say you have to have 2, but one of them has to do casework for 5 years before being a reviewer or can go out and testify. So it gets a little more specific with each set of standards that we follow. And here on this side, again, these are just some of the QAS requirements that we follow, how we write our reports. Proficiency testing, that comes up a lot in court. So proficiency test is a test that I take from an outside vendor. I process it like any other case, but I don't know the answer. The vendor does. So I process it. I issue a report to let them know, I think this is a 2-person mixture, and I think this person is included and this person is excluded. It's then looked at for grading to make sure that I'm doing my job appropriately and that all examiners are getting the same answers. All right, and emerging technologies, and I think I have flown through this, but that's good because we hopefully have time for questions. So rapid DNA testing, this is a fully integrated system capable of producing a DNA profile from a buccal sample or a cheek swab in 1 to 2 hours. These are used right now. There are some pilot studies in some states across the United States. Really, what we want to use these for is if you arrest an individual in Texas, we want to swab that individual's cheek, put it into the rapid DNA machine. In 1 to 2 hours, it's going to produce a DNA profile. It's going to compare that profile to CODIS, and you're going to find out that that person may have committed a crime in Florida. Then you can extradite or whatever you need to do on the legal side of it. But we're making sure that we're comparing to CODIS as quickly as possible to potentially hold someone in custody that may have committed a crime elsewhere. It's very user-friendly. There's very little interaction. You swab a person's cheek, you insert it into this machine, you insert a cartridge that has all the chemicals that you need, and you hit play. And it will do everything else in the background. It will do the extraction process that we talked about. It will give that DNA profile that we can then upload into CODIS to see if we get any hits. Something we're working on right now is next generation sequencing, or NGS. And this is to help us generate a lot of data in a little amount of time and a little bit of, and as cheap as we can. So right now to process a case, so typically a case of mine will have 5 to 10 DNA samples. In an ideal world where it's only my case and we're working around the clock, we can get results out in about 24 to 36 hours. That's just for my case. Well, I have 20 other coworkers that are examiners, and we all have about 20 cases on our desk at a time. So you're talking essentially 400, I think if I did the math right, cases in one day that could be processed. But if I'm only doing 10 or 15 at a time, that takes a lot of time. It takes a lot of resources. NGS may allow us to do 100 samples at a time. While it takes a little bit longer, it may take 3 days to complete those 100 compared to the 24 hours for the 10, it still lets us get a lot of information quicker and a lot more information. It's also known as massively parallel sequencing or ultra high throughput sequencing. And it's also going to combine kits. So when we talked about the Y DNA testing and the mitochondrial DNA testing and nuclear DNA testing, those are all different kits. Next generation sequencing will allow us to do it all in one chemistry. So again, it's a lot of data, and we can get a lot of information with that data. And it shows promise to improve degraded samples when we have really small amounts of DNA, and it will help with mixtures. So if we have a mixture of male and female, we might be able to split out that male and female a little bit better. And I flew through that, but does anybody have any questions? Anybody? Yeah.
That's what I'm saying.
And I forgot to, sorry.
I know you did. So has your STR mix ever been excluded in court, the use of it?
So ours, I believe, has not. I think we have 8 to 10 where it has been included. We've done Daubert hearings, and they've been included each time.
Have there been, other than FBI cases that have not been allowed into court, that are STR
I
mix?
think 1 or 2 across the community, yes. But a majority of them have been included. Do you have a follow-up?
Well, no one else? No, if there's someone else.
Okay. She's coming with the mic.
So in terms of the qualities of the specimens that you receive, could you also talk about the quality of the specimen as it's prepared and collected by the different agencies?
Yeah, sure. That's a good question. So I can't control what happens out in the field. However, and I can speak to the FBI, I know about our training program, but really what we're trying to do ultimately is not contaminate things. So when agencies are out in the field, we ask that they wear gloves, that they collect from one piece of evidence, package that, and then collect another piece of evidence after they've changed their gloves. Any PPE or personal protective equipment that they use, we also use that within the laboratory. As far as conditions of the item, so DNA is pretty robust, unless there's a few things that are pretty detrimental to it. High heat or UV. So a case that's been out in the Nevada desert for months, the DNA is probably not super great. But I always tell my agents, if you keep it cool, dry, and out of sunlight, it's usually pretty good for me to test. Bleach is detrimental. Coming off the heels of COVID, we all bleached down everything, including our groceries. I know I'm not the only one in here that bleached my groceries. But we use bleach for a reason, because it killed the DNA that made up COVID. Moisture is also bad because it's going to allow mold to grow, and mold will also be detrimental to DNA. So as far as out in the field, we keep it cool, dry, out of heat. It's good for us, but we can't control that. The things we can control are, again, collecting. So one thing that I've seen in trials is when they're doing a search warrant of a suspect's house, collect stuff from that person's house, change your gloves, change any tools that you're using before you collect maybe from a victim's house to make sure that there's no cross-contamination. Does that answer your question?
Preferences with regard to swabs, Dacron versus cotton?
So we did a study about that. We used to always use cotton. More recently, we've started using polyester swabs, and we found, so right now when we have felon in possession cases, we have firearms, and they're only charged as felon in possession. What we're doing is we're collecting from the firearm and we're sending it back to the field until they can get us a known sample so that we can actually compare. So we did a study swabbing guns, and we found that while both pick up a lot of DNA, the polyester seems to hold on to a little bit more. That doesn't mean that the cotton doesn't. So anybody that's out in the field, if all you have is cotton, that's great. And it's not a huge difference between the two. One's slightly better, so we choose to use the polyester. Wet versus dry swabbing, I see that a lot too. So that is where someone will take 2 Q-tips, one they'll add water to, the other one they won't. They'll swab an item with the wet swab and then go behind and dry it essentially with the dry swab. That doesn't really show any any difference either. And what we do in the lab is we actually combine those two swabs anyway. So if your location or your agents or your police officers or you go out into the field, the wet dry swab method doesn't really matter. Yes.
Are there limits to what you can amplify? I mean, it seems to me that if you can amplify something million, billion times, why can't you use the tiniest sample? So what's the limit of that?
Yep. So there are limits. And I think I skipped over because I think I actually removed it during the legal portion. So we do have limits. During our validation, and that's part of the validation process, we look at how much is too little that we can't amplify it. And I think right now we can go down. So ideally we're targeting a nanogram. So if you think of like an equal packet or a Splenda packet, a billionth of that packet is a nanogram. That's how much DNA we're ultimately targeting. That's an ideal situation. We can amp down to about 100 picograms, which is a tenth of that. When we looked at even lower than that, when we get to only 2 or 3 cells, that's not enough to amp. There are some, so low copy number, there are some laboratories that will process stuff low copy number. And what they will do with that is, knowing that this sample has really few cells on it, they will increase their amplification cycles. So each cycle, it takes your 10 copies and it makes it 20, and then it makes it 40, and it makes it 80, and so forth. We only do that 27 times, 27 cycles. Some laboratories might do 30 to take those few cells to try to get information that they can use. What happens sometimes with that is we get environmental, we call it drop-in. It's environmental DNA that just happens to, I don't like to say floating in the air, but it's picked up in such a small amount that we then start copying that, and we're not getting true results because we're getting this drop-in, which is sort of like a contaminant. So we can get very small amounts, but not super, super low. I need probably about 20 cells to get a DNA profile. Does that answer your question?
It does, because when we do the potions to consume,
Mhm.
we get those a lot.
Mhm.
The argument is always, well, why can't they, if they can amplify it, why can't they just cut the swab in half?
Yep. So the reason behind that is because we can't see DNA. And we don't know. So if I were to swab the top of this podium, maybe there's DNA, maybe there's not. So when I swab it, I don't know how much DNA is on there, if I even got any at all. It would be great if DNA turns like bright blue, and then you could tell this whole swab is bright blue. So when we ask to consume, we're going to take that entire swab and subject it to testing, because if I take half, I may have taken from the half that they didn't swab. Ideally, we tell our agents, when you swab, as you're swabbing, twist the swab so you get DNA all the way around it. But we don't know that's how it was collected. So we ask to consume the whole thing for our best chance to get any DNA that may be on that swab. It's very rare. I can only think of maybe less than a handful of times I've been told I couldn't consume. And usually what I do with that is I take as much as I can without consuming. So I'll usually take 3/4 of it. Sometimes you can tell the swab is matted, where it might have been rubbed on something or it's discolored. I'll take from that portion and leave some that the defense can test if they want. Yes.
I guess in terms of testifying as an expert witness, what are some of like the best practices or pet peeves you see from prosecutors when you're presenting this information? And like, how do you like to be prepped, I guess is.
Do you want me to be honest?
Yes.
So one of my biggest pet peeves is waiting to the last minute, which is funny because I'm a huge procrastinator and that's how I've gotten through my life. But a lot of times I don't expect a prosecutor to know this stuff. It took me years to become an expert in it. Some are really great, and that's great. But my biggest thing is just call. Let me talk to you first. Let me explain this to you. If you're not sure, you can explain it to me and let me tweak it if I need to. I have a set of standard questions that I give that I highly encourage prosecutors to use, but nobody has to. 95% of the time I'm taking up on that offer. And that really, just over my years of testifying, it really hits home the main points. I don't want to talk about statistics in super, super crazy depth to a juror. So my questions are very basic level. What is DNA? How do we get it? Sort of like this presentation was. So I tend to ask if they can use those. It lays it out really nicely for the jury. I had one prosecutor who was adamant that they've tried DNA cases many, many times and they were good. Okay, that's fine. I can think on my feet. And after they asked me to spell my name, the first question was, what were your results in this case? And I was like, well, let me back up a couple of things. So that's one of the things too, is just to work with your witness to figure out even what's important in the case. So if I don't have a case where I did serology testing, that blood or semen testing, we don't need to talk about it to the jury. Some prosecutors want to for one reason or another, but just an off the record conversation of what exactly do I have and how can I relay that to the jury? So that's probably my biggest thing. While we all pre-trials, every time I get one, I'm like, ugh, but I actually do like them because that's when I can speak freely without being on the record and without being nervous on the stand to figure out what are you going to ask? I know it can change, but is there anything crazy you might ask so I'm not stuck like a deer in headlights? Any more questions? Yeah, she's coming.
You may be familiar with the Avery case in Wisconsin because the FBI lab testified there. And there was questions about touch DNA.
Mhm.
And then after that case, I think studies came out, or maybe they were concurrent or before, showing that touch DNA could be transferred. Is that true?
You're absolutely right. And you just reminded me that I removed my transfer slides. So I will add them for future. So that is possible. So touch DNA, when we're talking about that, is DNA that's deposited through touch. So a lot of times agents will come through our lab and I talk to them on DNA and I describe to them that there's a difference to me, there's a difference between touch DNA and what we call wearer or handler DNA. If you were to swab the neckline of my jacket, I've been wearing it all day, you're going to get my DNA. It's close to me. There's some friction. If I were to touch this podium really quickly and swab here, I may or may not have left my DNA behind. That's what we consider touch DNA. Biologically, we all leave our DNA behind at various amounts and degrees. And there's no test that I can perform to say that I'm going to leave behind a cup at a 2-second handle and you might leave behind a half cup. I have no idea. It's not based on age or gender or anything like that. So with touching something, typically we don't like to do DNA testing on that because there's probably either something better in the case, not always, but we don't know. Are we going to run into that other issue where we only have a couple cells and we're not going to detect anything? Now, as far as the transfer, so there's 3 different types of transfer: primary, secondary, and tertiary. Primary is if I shake your hand and my DNA is now in your hand. Secondary, and I'm sorry, I'm using you as my example. Secondary would be me shaking your hand, you touching that door, and now my DNA is on that door. Tertiary is me shaking your hand, you touching that door, and you leaving, touching that door, and now my DNA is on your hand, where there's 3 steps of transfer. So touch DNA being transferred, that is possible because I expect the more times things are transferred, I would expect fewer and fewer cells to be transferred. Or sorry, the more times things are handled, the fewer and fewer cells would be transferred. A lot of times for juries, I like to use the example of paint. So if I had a bucket of paint and I put my hand in the paint, and then I went around to every chair in this room and did this, eventually there's going to be a chair that doesn't have any paint. Each chair is going to have fewer and fewer specks of paint on it. That's how I expect transfer to happen. So touch DNA or low levels of DNA can be transferred through or can be moved from one object to another while it's touch. Yeah, I could touch this podium. You could come touch this podium and get my DNA. That would be what we consider low number touch DNA, and that would be transfer. So it's possible. You're welcome.
Quick question, Amanda. So
Sure.
based on your likelihood ratio, can you talk a little bit about what language you would use in the report and in your testimony?
Yes, I can. So that's another thing, going back to the pet peeve question. Do I have it written out? Oh, I thought I did. Our language is very specific in what we have to say. So right now, our reports would say, let's say I have a likelihood ratio of 1 billion. I would say the DNA typing results from the swabbing of the firearm are 1 billion times more likely if Mr. Smith was a contributor than if an unknown, unrelated individual was a contributor. A lot of times what happens, so we prefer to refer to our report when we do that for a couple of different reasons. For one, it has to be said that very specific way. Statisticians have come to talk to us. Changing a couple words in that sentence could change the meaning of the sentence. None of us probably in this room would pick up on that, but you could have a statistician on the jury that said that's not exactly what she said. So a lot of times what I like to do is talk to my prosecutors and say, does your judge have a problem with me referring to my report? When I get into the numbers, septillion, non-million, it's 427 septillion, and it's 4 people, I don't want to mess up anything. I think only once have I been told I couldn't refer to my report. So then that gets a little dicey. Also, a lot of times I don't have just one statistic that I'm giving in my report. It could be 8 different pieces of information with 4 people compared to each of them. That's essentially could be up to 32 likelihood ratios that I'm giving. A lot of times I hear the prosecution fallacy. So what they'll do is they'll combine the random match probability and the likelihood ratio. So they'll say, well, she said the likelihood ratio was 1 billion. There's 8 billion people in the world. So I would expect the likelihood ratio is 1 in a billion. So I'd expect 8 people to have this profile. That's the random match probability. Where I like to use the world is trying to describe to a jury what exactly a non-million is. Usually I just like to use the number of zeros. If I tell them 1 billion is 9 zeros and a non-million is 30, they will understand that. We also have sometimes where prosecution or defense will invert the likelihood ratio, and they'll say it is 1 billion times more likely he did it. No, I can never determine if someone did it or did not do it. So there's a very specific, I wish I would have had an example here, but the way I said it before, it's a very specific way. It's an if-then statement that I have to give. So we prefer to go based on our report. Any more questions? We're all DNA experts now. Well, I appreciate the time. My contact information, I think you got some speaker information. If not, here is my phone number and my email. Feel free to reach out to me. I truly do like talking to the field. Even if you're thinking about trying a DNA case and you're like, I'm not really sure what I have, certainly let me know. I can walk you through it. Some people have asked for my questions that I give. I would be more than happy to give them to anybody in the room. Just email me. Again, you can change them for your expert, change them for your case. That doesn't mean mine are the best. But some people, if you haven't tried a DNA case, at least it's a starting point. So thank you so much for your time. And I hope you got a little out of it. I know it's a lot of science and math heavy. And I know I buzzed through that. I was hoping to make it closer to the hour and a half. But hopefully if you have a DNA case, you can kind of slow down and grasp this a little bit more. So thank you so much.